Welcome back to another deep dive. We are just so thrilled to have you joining us today.
It's great to be here.
Yeah, and today's mission is, well, it's a really big one. We are unpacking this highly technical weekly intelligence bulletin. It covers global microbiology, biotechnology, and fermentation developments, specifically looking at the data from late February to early March of 2026.
Right.
And if you've ever actually glanced at one of these reports, you know they're usually reserved exclusively for, like, bioprocess venture strategists. They are incredibly dense.
Oh, absolutely.
I mean, we are talking pages and pages of jargon.
Oh.
But that is exactly why we are here. Our goal today is to completely cut through that noise, bypass all that information overload, and extract the real, tangible, bench-to-tank stories for you.
Yes.
Think of this as your ultimate shortcut to being perfectly well-informed on the absolute cutting-edge future of synthetic biology.
It's a vital conversation to have right now, honestly, because these bulletins aren't just academic exercises. They literally map out the future of physical manufacturing.
Right.
What we're looking at today isn't just about what's happening in highly controlled, pristine lab environments from Boston to Shenzhen to Singapore. It's really about the why.
Yeah.
Specifically, we're looking at the often brutal, unforgiving reality of scaling biology, taking a fragile organism that performs perfectly in a tiny, perfect glass petri dish.
Right, where everything is optimal.
Exactly. And forcing it to survive and produce in a massive, turbulent commercial steel tank, which is arguably one of the hardest engineering challenges of our times.
Okay, let's unpack this, because the first section of our sources is all about giving these microbes what looks like some serious biohacking upgrades just so they can survive those harsh conditions.
Yeah, the metabolic engineering side.
Right. The bulletin highlights some massive breakthroughs there. But before we get into the numbers, I want to talk about the concept. There's this fascinating preprint out of MIT's Prather lab. They've been using a refined CRISPR system. CRISPR-Cas9 in E. coli. And the way I'm reading this, it's essentially like giving these bacteria advanced elite-level stress management training so they don't just shut down when the environment gets tough. How exactly are they doing that?
It's a really great analogy. In a commercial bioreactor, bacteria experience extreme oxidative stress. Basically, it's chemical damage to their cells.
Ouch.
Yeah, it's rough. So the MIT team used CRISPR to stabilize the bacteria's internal metabolic pathways under those stressful conditions. And the results they published are wild. Because the bacteria weren't shutting down from stress, they achieved a massive 2.3-fold increase in their titer.
Let me pause you right there just to make sure we are all on the same page. When we talk about titer in these bulletins, what exactly are we measuring?
Simply put, titer is just the concentration of the product you actually want floating in the liquid of the tank.
Okay, got it.
So in MIT's case, they were trying to produce a chemical called mevalonate. Thanks to the CRISPR upgrades, they hit 18 grams of mevalonate per liter of liquid. And they pushed their production rate to 0.75 grams per liter every single hour. What's fascinating here is how that connects directly to the broader industrial puzzle. That MIT research works beautifully in tandem with another major development in the bulletin from Novo Nordisk.
Oh, the yeast thing.
Yes. They recently filed a patent for a synthetic terminator library in Saccharomyces cerevisiae, which is just the scientific name for common baker's yeast.
Wait, so the exact same yeast we use to make sourdough bread and beer is getting a massive genetic upgrade.
The very same.
It's like giving baker's yeast a microscopic suit of armor.
Precisely.
Yeah.
Novo Nordisk integrated this new genetic library with carbon dioxide and hydrogen feeds. And they achieved an incredible ethanol tolerance of 145 grams per liter.
That's huge.
It is. That is a 15% improvement over wild natural yeast. And the why here goes back to your point about stress management. Industrial tanks are violent environments.
Right.
When you scale up, you introduce extreme pH swings and toxic buildup of the very product the cells are making, in this case, ethanol. These genetic upgrades act like internal armor, solving the fundamental disconnect between the delicate, coddled conditions of a university lab and the physical realities of industrial manufacturing.
And from what I'm seeing in the sources, it isn't just about making them tougher, it's about what we are actually feeding them.
Feedstocks, yeah.
Right. There's a trial in Singapore that caught my eye because it sounds like literal science fiction. They quite literally fed plastic specifically, a broken down form of pape plastic, to a type of bacteria called Pseudomonas.
Yes, the peaky hydrolysate.
Exactly. And the bacteria turned that plastic waste into a biodegradable bioplastic called PHA. I mean, we are feeding our old plastic waste to microbes to make better, cleaner plastics, but is that actually efficient on an industrial scale?
It is becoming incredibly efficient. In that Singapore trial, they achieved a production rate of 0.55 grams per liter per hour. Financially, that slashes operational expenses by 45% compared to feeding the bacteria traditional expensive sugars like glucose.
Wow, 45%.
Yeah, and the European data in the bulletin echoes this entire pivot away from traditional agriculture. A German study linked to the European Food Safety Authority used what we call e-feedstocks.
E-feedstocks, like electronic feedstocks.
Sort of. It means feedstocks derived from captured carbon and renewable energy rather than grown in a field. In this case, they took captured CO2, converted it into formate, and fed that to Cupria fidus bacteria.
Okay.
By doing this, they boosted the yield to 0.72 grams of product for every gram of formate they fed it. What you are seeing is a global shift. Relying on agricultural sugars is expensive.
So true.
By engineering strains that can eat plastic waste or captured carbon, the industry is trying to push their running costs down below $2 a kilogram. Though, as the bulletin warns, when you use municipal waste streams, you do increase your contamination risks by about 15 to 20%.
Here's where it gets really interesting. Because you can have the toughest, most efficient plastic-eating bacteria in the world, but if you can't scale it up from a tiny glass flask to a massive commercial facility, it doesn't matter.
It means nothing.
Exactly. And that brings us to what the biotech world calls the valley of death, the physical reality of scaling up. Our sources detail a pretty brutal failure out of Shenzhen last week.
Oh, yeah.
A company tried to scale their process from a small 10-liter pilot tank to a 1,000-liter commercial fermenter, and their yield completely crashed. It fell by 45%. The bulletin mentions they ignored the physics of sheer stress. Why is scaling up a liquid so difficult?
It is a classic trap, and it all comes down to the scaling laws of physics. As your tank gets larger, mixing the liquid evenly becomes exponentially harder. To keep the bacteria alive, you have to pump oxygen into the water.
Right.
The metric the bulletin uses for this is the K-law value, which is basically just an engineering term for how fast and how well you can dissolve oxygen gas into a liquid.
Okay, so a higher kilo value means your bacteria are breathing easier.
Exactly. But to get a high Kla in a massive 1,000-liter tank, you have to spin the impellers, the mixing blades, incredibly fast. The bulletin details a run by Novartis that perfectly illustrates this physical trap.
Oh, I read about this.
Yeah. In their massive tank, the mixing time exceeded 15 seconds, and the tip speed of those giant steel blades went over 1.5 meters per second.
Which sounds like putting these highly engineered microscopic organisms into a giant industrial blender.
That is exactly what it is. That sheer physical force literally tears the cells apart. In that Novartis run, cell viability dropped by 22% just from the physical trauma of the blade.
That is wild.
And if the blades don't kill them, the thermodynamics might. A Dutch scale-up study showed that at high cell densities, they couldn't pull the heat away fast enough. The bacteria were generating so much energy that they experienced temperature spikes of 4 degrees Celsius.
Wait, if you have a massive 1,000-liter steel tank, how do you even go about cooling that down fast enough?
That is the core problem. In a small 10-liter tank, you have a lot of surface area relative to the volume of liquid, so heat escapes easily through the glass.
Right, that makes sense.
But in a 1,000-liter tank, the volume of liquid is so vast that the cooling jackets on the outside simply cannot chill the center of the tank fast enough. For a highly engineered microbe, a sudden 4-degree swing is catastrophic.
So they just bake themselves.
Pretty much. In the Dutch study, that heat spike reduced their overall production rate by 18%. When you're an investor reading these bulletins, you have to realize that physical limitations, heat transfer, mixing times, oxygen delivery will ruthlessly dictate your biological success.
But the hardware engineers aren't just sitting back and accepting defeat. I mean, if the physical hardware is maxing out, they're finding ways around the physics problem. To solve that oxygen and blender problem we just talked about, Shenzhen's BioEquip just unveiled a new bioreactor called the Oxymax 5000.
Right, the microbubble system.
Yeah. Instead of violently stirring the tank with giant blades, They use microbubble sparging. They are basically injecting microscopic bubbles of oxygen directly into the mix, hitting an oxygen transfer rate, that CLR value we mentioned, 450, while reducing energy use by 32%.
It's a brilliant pivot. And there's another approach in the bulletin that completely bypasses the liquid physics problem altogether. An MIT spinout entirely ditched the liquid tank model.
Oh, the solid-state fermentation.
Yes, they grew Aspergillus fungi directly on solid agricultural waste using precise humidity control instead of submerged liquid mixing.
So no giant blender blades, no liquid cooling problems.
Exactly. And the numbers prove the concept. They achieved a massive yield of 0.65 grams per gram of substrate. For context, the traditional liquid submerged methods usually max out around 0.48.
That's a huge jump.
They literally changed the physical environment to beat the physics problem. Pushing those efficiencies is the only way the industry will ever drive costs down enough to compete.
That perfectly transitions into the next major theme of our deep dive. If physical hardware is reaching its limits, is there another way around the valley of death? The answer seems to be software. The phrase, software eats biotech, has never been more accurate. Let's talk about the AI and digital twin technologies the Bulletin covers.
This is where it gets really futuristic.
Definitely. There's a new open source tool called FluxSim version 3.2. What exactly is this doing for these bioengineers?
FluxSim is an AI-driven pathway prediction tool. Before a scientist ever edits a gene in a real lab, they use software to map out how the bacteria's metabolism will react. The big news in the bulletin is that FluxSim version 3.2 is now outperforming the older industry standard, a program called Cobra Bee, by 22% in accuracy.
And the accuracy of those models leads directly to what the Bulletin calls digital twins, right?
Yep.
I was reading about Siemens' new BioTwin version 4. From what I understand, a digital twin is an exact, highly complex software replica of your physical bioreactor and your biological organism. It's like a video game simulation of your factory.
That is a perfect way to visualize it. Siemens used this BioTwin software to model a transition from a small 10-liter tank up to a 1,000-liter tank. By making tweaks entirely in silico, meaning inside the computer simulation, they predicted the final real-world yield within an 8% margin of error.
That is incredible.
For you, the listener, understanding the impact of this is crucial. Founders can now perfectly simulate the physics of a massive commercial tank, watch their virtual cells die from sheer stress, fix the impeller design in the software, and completely de-risk their scale-up. It is estimated to cut those valley of death failures by 40% before a company ever spends $500,000 on a physical pilot run.
And it isn't just pre-planning. Software is managing microbes in real time while they're actively in the tank. There is a fascinating study from a Singapore-MIT collaboration looking at bacterial biofilms. They found that under high-density conditions, Pseudomonas aeruginosa bacteria experience severe osmotic stress. The bulletin says the bacteria literally panics.
They do. It's a profound stress response. To stop itself from completely crashing, the bacteria reroutes 40% of its entire metabolism away from making the product you want, and instead uses that energy to produce a protective slime called alginate.
Oh, wow.
Yeah, that microscopic survival mechanism causes an 18% loss in your overall yield.
But if you can see that stress happening in real time, you can intervene. Which brings us to the new sensor technologies. Bosch just released an inline Raman probe. It acts as a real-time window into the bioreactor, right?
Exactly. In the past, you had to pull a sample out of the tank and take it to a lab to see how the cells were doing. This Raman probe senses actual biomass and metabolic states in real time, directly inside the tank, with 5% accuracy.
And they pair that with AI.
Yes. When you pair that sensor with machine learning software, the computer can predict when the cells are about to panic and automatically adjust the nutrient feed to calm them down. The bulletin notes that in Chinese hamster ovary cell cultures, this real-time AI intervention lifted the final titer by 28%.
Okay, so with yields going up, costs coming down, and software de-risking the massive physical failures, the financial markets are absolutely taking notice. The Bulletin's market intelligence section shows some jaw-dropping numbers. Following the money here is wild.
The capital flowing in is unprecedented.
Venture capitalists poured $2.1 billion into the biotech sector in just one single week. But here's the critical detail. They aren't betting on single products. $1.4 billion of that funding went strictly to platform technologies, like those AI-strained design tools we just talked about, while only $700 million went to specific products like oncology drugs.
Investors want discovery engines, not just a single winning lottery ticket.
Exactly. And the reason they are pouring money in is because synthetic biology is starting to aggressively eat traditional markets. Ginkgo Bioworks has a latest pilot producing isoprene at 95 cents a kilogram. Traditional petrochemical isoprene costs $1.20 a kilogram. They are successfully undercutting the fossil fuel industry.
It's a massive shift.
And DSM is producing synbio vanillin at $8 a kilogram, which is a direct threat to the entire traditional vanilla bean farming sector.
This raises an important question, though. When you start displacing $100 billion legacy chemical and agricultural markets, governments and regulators sit up and take notice. And as these synthetic biology platforms accelerate, the regulatory hammer is coming down hard on the safety of these engineered strains.
Yeah, the FDA updates.
Exactly. The FDA has just updated their good manufacturing practice guidelines with a new safety by design mandate.
What does that actually mean for a biotech startup?
It means a massive increase in compliance. The FDA is now requiring whole genome sequencing for all microbial strains to perfectly map any contamination risks. You can't just say your bacteria is safe. You have to map every single letter of its DNA to prove it.
Wow.
Meanwhile, in Europe, the European Medicines Agency's Novel Foods draft has tightened regulations on precision fermentation. They are demanding absolute host strain characterization via next-generation sequencing, along with intense allergen profiling and strict limits on endotoxins.
Right, and the bulletin makes it clear this isn't just paperwork. If biotech founders aren't building this intense DNA sequencing into their process from the very beginning, what happens?
They are facing massive regulatory delays of 6 to 9 months before they can get approvals. It's a huge bottleneck that can bankrupt a startup if they aren't prepared to navigate it.
And navigating the reality of this industry, separating the hard science from the hype, is practically a full-time job. Which brings us to a viral news story that blew up the internet last week. You probably saw this. It generated over 50,000 engagements online.
Yes, the CO2 fermentation company.
That's the one. A startup out of San Francisco claimed they achieved zero-cost feedstock by using atmospheric carbon dioxide fermentation. They promised they were producing protein at just 10 cents a kilogram, and they claimed they could offer an instant scale-up right to a 10,000-liter facility. It sounds like the silver bullet to solve global hunger and climate change in one fell swoop. But is it real?
Unfortunately, when the Bulletin ran the numbers on their claims, the math just didn't align with the physics. To produce 1 kilogram of protein from carbon dioxide, the basic mass balance requires an input of 1.8 kilograms of CO2. Furthermore, the energy required for the biological fixation of that carbon is massive, about 15 megajoules per kilogram.
To put that in perspective for you, 15 megajoules is roughly the same amount of energy it takes to run a standard household electric oven on high heat for an entire hour, just to make 1 kilogram of protein.
Exactly. So even if the feedstock is literally free air, the electricity and energy costs to run the process will push operational expenses to at least 80 cents a kilogram at scale, not zero.
Wow, so the core premise is flawed right out of the gate. But what about their yield claims? They reported a yield of 0.75 grams of protein per gram of input.
This is where it defies what we call the Gibbs free energy mismatch. To put that simply for our listeners, it violates the basic accounting of physical chemistry. You literally cannot get more energy out of a chemical reaction than you put in. The absolute theoretical maximum yield for this type of carbon fixation is only 0.55. Claiming 0.75 is scientifically impossible with current biological pathways.
And what about their claim of an instant scale-up to 10,000 liters?
As we discussed earlier with the Novartis example, that completely ignores the physics of mixing time. Jumping straight to 10,000 liters means your mixing time will jump from 5 seconds in a pilot tank to over 30 seconds, which risks a massive loss in production rate due to poor oxygen transfer.
The cells would just suffocate.
Right. So the analytical takeaway from the source material is that without hard pilot data, this appears to be a venture capital pitch rather than a market-ready reality. The realistic cost of goods sold for a process like this, post-scale, is likely much closer to $1.50 a kilogram, not the 10 cents they are claiming.
So what does this all mean? If you take a step back and look at the journey we've just been on, from the CRISPR-edited bacteria getting stress management training at MIT, to the sheer physical trauma of cells in a 1,000-liter commercial blender, right down to the AI digital twins predicting the future and regulators demanding complete DNA maps. The core takeaway for you today is that the true magic of synthetic biology isn't just happening at the genetic level on a lab bench.
It really isn't.
The real revolution, the real challenge, is surviving the brutal physical scaling laws, the harsh thermodynamics, and the stringent economics of the commercial bioreactor. Being able to edit a genome is amazing, but engineering an entire industrial bioprocess that can actually outcompete the petrochemical industry, that is the real frontier.
It truly is. And it leaves us with a provocative thought to consider regarding the long-term impact of this entire field. We talked about synthetic vanillin being produced in a bioreactor at $8 a kilogram. If these massive physical scaling challenges are eventually solved by AI and next-generation bioreactors, what happens to the global agricultural economy? When synthetic biology scales perfectly, it won't just replace fossil fuels, it will replace farming. Millions of traditional farmers, like the generations of families growing vanilla beans in Madagascar, could suddenly find their entire legacy obsolete, outpriced by a steel tank in Shenzhen or Boston. If tools like Siemens Biotwin and Fluxim can accurately predict physical scaling failures and perfectly optimize metabolic pathways in a computer, how long until the biggest biotech breakthroughs happen entirely in silico?
That is wild to think about.
Are we rapidly approaching an era where new synthetic organisms are fully designed, stressed, and scaled in a digital universe before a single cell is ever cultured in the physical world? And if we finally master the physics of the commercial bioreactor, are we truly prepared for the massive socioeconomic earthquake that will inevitably follow in the soil?
That is a staggering thought to leave off on, and definitely something to keep an eye on as this technology continues to scale. We want to thank you so much for joining us on this deep dive into the absolute cutting edge of bioprocessing. As always, keep questioning the hype, keep looking for the hard science, and we will catch you on the next one.
Hi there, thank you for staying with me until the very end. If these technical insights help bridge the gap in your own process, please hit subscribe and make sure to hit the notification bell. Stay connected for our next deep dive into the realities of biomanufacturing. I am Prasad Ernala. Thanks for listening.